everyone. So we have a video on how you can draw with microbes. So for this video, we use Photobacterium phosphorium, which is, as you can see in this picture, quite luminescent in the dark. It glows this nice kind of greenish, bluish color. So in this picture, it was just the cultures just in a dark room that I took a picture of, and I thought it looked really cool. In microbiology, there's a lot of microbiologists who just in their free time like to make agar art. They kind of take plates and they streak bacteria onto the plates to form really amazing designs. Some of them are really, really beautiful. And you can use like bacterial cultures that have specific colors or even textures to make the different designs. And there's also ways to mess with the genomes of bacteria to make them fluoresce a specific color. So you can use either naturally occurring ones, like the Photobacterium phosphorium that we used, or artificially colored bacteria. And if you want to see lots of examples of the agar art, you can go to the ASM website and look for the agar art there. I think they have a competition every year for the best agar art that their microbiologists can create. And so a little bit about the Photobacterium phosphorium. It's an organism that's found on marine fish, and it forms this luminescence as it's an oxygen-dependent reaction. So in this picture, which we took from the dark room in lab, you can see in a flask, oxygen is only going to be at the top level of the flask, and so the bottom part of the liquid culture is going to be anaerobic, or there's not going to be any oxygen there. So just this small band at the top of where the oxygen is going to be lighted up, and so you can see that in this picture. So this organism lights up just in the dark. It doesn't require any special lights or fluorescence to see the light that's emitted from the organism. This organism is also psychrophilic, so that means it likes the cold. So when we incubated the bacteria for this project, we didn't put it in an incubator or anything. We actually put it in the refrigerator. So to make our agar art that you'll see in a little bit, we first had to make the paint or the solution to streak onto the plates with our swabs. So to do that, we took some of the culture medium first and put it in some vials, and then picked up from the plates some parts of the colonies of Photobacterium and mixed it into the culture medium vials. And then to make the letters that you'll see in a little bit, we first filled out the whole plate with bacterium. So we took a swab and covered the whole entire plate with our paint, so to say. And then to make the designs, as you can see here, we drew on some butcher paper, or Emma did, our president drew on butcher paper design, and then we took plates and placed it over the design to fill it up, and then we traced over with our culture medium to fill out that design. And then we waited for 24 hours for the microbes to grow, and then we came back the next day to take the pictures. And so here you can see the pictures that we took. In this first one, you can see our design in the light when it's not emitting, or when we can't see the luminescence that the organism is emitting. And then you can see it's emitting some as the light goes down, and then in the dark you can really, really see the luminescence of these bacteria and the beautiful designs that our president created. And so here's our first design, a coronavirus, because that is part of the time that we are living in. And then we have a collection of microbes of various kinds. It looks like an E. coli and a chain of them and some little clusters of microbes. And then we have a very nice microscope I love to look at microbes under the microscope. And then, as always, go Ags! And we would like to send sincere thank yous to the professors that helped with this project. Dr. Martin helped with the Photobacterium cultures and advised Emma and myself on how to form the Agar art and helped us with quite a bit of it too. And then Dr. Bragg helped us take the pictures. And we'd also like to send a huge thank you to the prep staff for putting together all of the lab equipment for us to use and just getting things ready for us to be there. Thank you so much.